So our learning objectives for this video are to understand the purpose of DNA replication and the process of DNA replication. When I say DNA replication, another word you might hear is DNA synthesis. And synthesis means making. It's simply making DNA. It's the same thing as DNA replication. The enzymes involved, how the copies are made is pretty important to the idea of DNA, and limitations of that process. So our goal here, as I kind of mentioned in the previous video, is to make a second copy of the DNA molecule, hopefully one that is exactly the same. So for that we need DNA strand, we need enzymes, and we need nucleotides. Now, when would we be doing DNA replication? As I suggested in the previous video, whenever we grow new cells, whenever they divide by mitosis, which we will see coming up soon, we'll need to copy the DNA so that we have a copy for each of the new cells. And similarly, when we get ready for meiosis, for reproduction, producing the germ cells, sperm and eggs, we also need to make a copy of the DNA, because of course we ourselves need to retain a full set of our DNA, and we also need to send a set of that on to the new cell or the new individual. So basically the process here, summarized, you'll remember this similar diagram from the previous video. And remember that we have the 3' end down here, you have the 5' end here, and it's anti-parallel, so the other strand is 3' at the top, 5' at the bottom. So the first enzyme that's involved is helicase, and the job of helicase is to unzip the DNA strand, break those hydrogen bonds, and separate the two strands from each other. And then what will happen is, in making a new strand, we will start with primase enzyme carries in an RNA primer. And this is just an RNA nucleotide, and it's temporary. It will leave at the end. Because it's RNA, it can't become a part of the DNA molecule. But we need that because DNA polymerase here is going to add nucleotides, but it can only add nucleotides to an existing nucleotide. We need primase to start the strand and let DNA polymerase continue that, because DNA polymerase needs to build phosphodiester bonds, those covalent bonds between the nucleotides in the same strand, and there's nothing to add a covalent bond to when there's nothing there. So now we're going to add in nucleotides, and not just any nucleotides. We're adding in the complementary nucleotides because remember we still have these base pairing rules. The T must pair with A, and C must pair with G, and so on. So you'll see that this new strand here in gray is the same as the old strand that was separated from it. So we've made an exact copy here, and the same thing would be happening on this strand, just in the opposite direction. So we would have a primase starting up here, and we'd form a new strand here. And you can see that the primer disappeared now. As I said, it was temporary. So in the end, we have the template strand in white was the original strand, and the daughter strand in gray is the new strand. So we've made, well, we started with one strand, and now we have two strands, but the way that we did that is by using one half of the old strand on each new molecule. So that is something called semi-conservative. It means that each new molecule contains half of the original and half new. Of course, the alternative is that we make, from the original two-stranded molecules, we make a completely new strand where both would be gray, but that's not the case. And the work of helicase is working at multiple points along the DNA strand. Remember, for example, chromosome 1 in your cells is 247 million base pairs. That would take a very long time to replicate in one direction. Kind of like if you're building a long railroad, the most efficient and quickest way to do that is not to start at the beginning and work towards the end, but to have crews working at multiple positions in both directions, and so they'll meet in the middle each time. And so that would be the quickest way to build your railroad, and that's what happens with DNA replication, because, as I've said, the cells are pretty efficient at what they do. So there will be helicase enzymes working at each of these green arrows in the diagram, and new strands will be formed in gray here at each one. And eventually these bubbles, and that's what it actually looks like on an electron microscope picture, you'll see these bubbles on the DNA. Eventually they'll join together and you'll have the continuous new strands. Okay, now let's imagine this situation where we've got this strand here, and that's the 5' end, meaning that the new strand on the other side will then be a 3' at this end, and 5' down this way. Now, DNA replication can only go from 5' to 3' because DNA polymerase can only add onto the phosphate end of the previous nucleotide. So that means that this one can get synthesized in this direction. This strand needs to go in the opposite direction. It cannot be synthesized this way. And remember that we've got helicase working in this direction. It's working continuously and opening up that replication fork. So now as we grow that strand, you can see that this bottom strand has to be made in chunks, whereas this one on top can be made continuously as this fork moves to the left. So I'll watch that again. It's this strand on the bottom that has a problem because it needs to be made from left to right, away from the replication fork. And so as new template strand becomes exposed in this area, we can make a new strand from left to right. And there we go. So this strand here is called the leading strand, where it's made continuously, and This one is called the lagging strand, this new strand, because it's made in chunks called Okazaki fragments. And there we go. That leading strand is this strand that was made continuously on this diagram from right to left. The other one is called the lagging strand because it's made away from the replication fork. Remember that on this one we had 5' here and 3' here. These pieces, there's 3 of them in the diagram, are called Okazaki fragments because they were discovered by a guy called Okazaki. Now these new ones need to get glued together with something called ligase. And they're just going to take the pieces of the Okazaki fragments and glue them together into one continuous strand. And that's, of course, another enzyme. So we've seen 3 enzymes here so far. We've seen helicase, 4. So we've seen 4 enzymes here so far. We've seen helicase, primase, DNA polymerase, and ligase. So DNA polymerase is not perfect at its job. There's limitations to the functioning of that enzyme.
So think about those two things, and we'll take a look at those in class and discuss that further.
So in review, each new DNA molecule consists of one old strand and one new strand, called semi-conservative. Helicase unzips the DNA strand, primase adds the RNA primer, DNA polymerase adds the complementary nucleotides, and that part's important. Complementary refers to the base pairing rules, so the complementary nucleotide to thymine is adenine, in the 5' to 3' direction. And then replication of one strand is continuous and the other is in fragments. And that's it for this video. In class, we'll have a chance to, first of all, make a model of DNA and see how it works a bit better with replication. And second of all, we'll have a chance to look at your own DNA in solution, where we take a whole bunch, and we'll take a look at your own DNA, where we take a whole bunch of DNA, break it up out of the cells, so we can see it with our naked eye, which is just kind of neat.